Good morning, all you Sioux-High AP Biology students. Shake those cobwebs off. It's 5:45 a.m. and time to get the day started. Today we will continue our discussion of proteins by introducing the idea of protein synthesis. So get your notes out, something to write with, and let's biosynthesize. Okay, recall that proteins are the most important of all the organic molecules that make up living organisms, and that proteins are long chains of amino acids in a specific sequence. Remember, they're bonded by what's called peptide bonds, and thus they're also called polypeptides. The question is, how does this happen and where? So let's take a look at this process called protein synthesis. Write in your notes that protein synthesis is so important, it's often referred to as the central dogma of life. Remember that the word synthesis means to build or put together. So for instance, we talked earlier about dehydration synthesis, which is the process of putting together all of each of the four organic molecules, and you now know what those are. Remember, proteins, carbohydrates, lipids, and nucleic acids. So we're exploring the entire process of making proteins. What we mean by that, and what I'd like you to get in your notes, is from gene to protein. So from the gene that has the instructions from amino acid sequencing to making the actual protein in the ribosomes is where we're going. So let's start by having you get the diagram in this slide in your notes. So go ahead and do that right now. Make sure you label it correctly. Now, as you can see, this process involves three important steps. So DNA replication. Remember, replicate simply means somehow to copy DNA. So you might want to get that in your notes. Replication means we got to copy DNA. Transcription. So step 2 is transcription, and step 3 is translation. So get those three processes in your notes, and then let's explore these processes in some detail to prepare us for some exam questions that will ultimately appear. Okay, now first a general overview of protein synthesis, and then we'll get into some detail about each of the processes involved. First, write in your notes that the information content of DNA is in the form of specific sequences of nucleotides along the DNA strands found in the nucleus of a cell. And remember, chromosomes are made of DNA, so that's what we're talking about, the DNA in chromosomes of a cell. Each sequence of nucleotides, write down in your notes that each sequence of nucleotides codes for an amino acid. So each sequence of nucleotides codes for an amino acid. Now, do you remember the basic structure of a DNA nucleotide? If you said a 5-carbon sugar, a phosphate, and a nitrogen base, you'd be correct. And whenever you hear the word nitrogen base, you think of adenine, guanine, cytosine, and thymine, and then we're going to introduce uracil into the equation today. Now, also write that the DNA inherited by an organism leads to specific traits by dictating the synthesis of proteins. That's really important. Now, recall that a protein is a sequence of amino acids, right? And that the order or sequencing is determined by the DNA you inherit from your parents. Now finally, get in your notes that the process by which DNA directs protein synthesis and gene expression includes 3 stages: replication, transcription, and translation. Now remember, the protein will ultimately end up being constructed in the organelle called the ribosome. So when you were in 9th grade biology and they ask you what the function of a ribosome was, you said protein synthesis. Well, now you know that what that really means is that that's where proteins are assembled. Now, in order to get them assembled, two things have to happen. The instructions for the sequencing have to get from the nucleus to the ribosome. And then we've got to find the amino acids somewhere and get those to the ribosome so that the two can meet and we can put those together in the correct order. That's where we're heading. Okay, so next write that cells are governed by a cellular chain of command in the following order. So you might want to write these as a flow chart with arrows in between. So DNA, and we'll say DNA replication or copying, RNA transcription, and then translation into a protein. So those are the three steps: DNA replication, RNA transcription, and then translation into a protein. So here you see the primary sequence of a protein now. So you should also recognize that these circles now are sequences of amino acids. So that's how you know it's a primary structure. Now it's actually folded a little bit, so you could make a case that it's on its way to secondary structure, but this is still in its basic form, which we call primary structure. Now get in your notes that transcription is the synthesis or building or putting together of RNA under the direction of DNA, and that it produces something called messenger RNA. Messenger RNA is also shortened to mRNA. Then write that translation is the actual synthesis or building of a polypeptide or protein, which occurs under the direction of mRNA and occurs in the ribosomes. So DNA and messenger RNA become the directors. So again, we're talking really basic right now, and then we're going to get really specific here in a bit. But it's important to know that DNA directs the construction of messenger RNA, and messenger RNA supervises the ultimate construction of a protein. And we're going to find that messenger RNA's job is to supervise a bunch of other RNAs that get synthesized in this process as well. So get ready to list those and their functions here soon. Now let's do a quick review of the similarities and differences between RNA and DNA before we move on. So I'd like you to get this Venn diagram in your notes. So you've done Venn diagrams here recently. Get this Venn diagram in your notes right now so that we can refer back to it during the podcast and when we're talking about it. So let's quickly go over the similarities first. So that would be right here in the middle of your Venn diagram that you've just constructed. So each has nucleotides, meaning a phosphate, a 5-carbon sugar, and a nitrogen base. Now, it's worth mentioning or introducing right now that the 5-carbon sugars are different. Ribose is the 5-carbon sugar in RNA, and deoxyribose is the 5-carbon sugar in DNA. So it might be worth writing that down right now. Both of them have 4 bases. Now, they're not the same 4 bases, but they do have 4 nitrogen bases. It might be worth writing that RNA is adenine, guanine, cytosine, and uracil, where DNA is adenine, guanine, cytosine, and thymine. And they're both chain-like molecules. In other words, you've seen a model of DNA. It's a long chain of nucleotides called a polynucleotide. So that makes them similar as well. So that's 3 structural similarities between RNA and DNA. Okay, now let's go over the differences. RNA uses the nitrogen base uracil. We just mentioned that instead of thymine, with all the other 3 nitrogen bases the same. Now this makes sense. Different structure, different function, right? So because RNA and DNA have different structures, you can imagine that they would have different functions as well, and you'd be right. DNA is a very long molecule compared with a relatively short RNA molecule. So DNA long, RNA short. And we just mentioned that the sugars are also different. Deoxyribose is the sugar in DNA, ribose is the sugar in RNA, and obviously the difference is deoxyribose lacks an oxygen atom. So DNA uses the 5-carbon sugar deoxyribose, meaning it lacks an oxygen atom, while RNA uses the 5-carbon sugar ribose. So those are your similarities and differences between DNA and RNA. Now we might also say location could be a difference. DNA never leaves the nucleus, so you might want to write that down. DNA never leaves the nucleus, whereas RNA does, especially messenger RNA. That can function in the nucleus and outside in the cytoplasm as well. So there's another difference between the two that you should know. Now, it's also important to remember that proteins need to be made in all organisms, both prokaryotes and eukaryotes. Remember, a prokaryote is something like a bacteria. It has a single chromosome, no nucleus, whereas a eukaryote has multiple chromosomes, therefore they're housed in a nucleus. Recall that prokaryotes only have a single chromosome, don't need a nucleus, whereas eukaryotes fully formed nucleus with multiple chromosomes. So if you don't know that difference or you can't remember it, you might want to jot it down in your notes again. Now I want to emphasize that because these structures are different, you can expect that their function will be as well, and that the process of protein synthesis will vary slightly between prokaryotic cells and eukaryotic cells. Now, write in your notes that because in prokaryotes transcription and translation occur together, meaning messenger RNAs produced by transcription is immediately translated without any additional processing. So that all happens at the same time in a prokaryote. That is not true in a eukaryote. Now, this was a short answer recently on an AP exam. It asked you to explain the difference between the protein synthesis in prokaryotes and eukaryotes. Here's your answer. In prokaryotes, single-step transcription and translation because of a single chromosome, whereas in eukaryotes, multi-step transcription and translation because they're in a different location. So there's your differences. Now, in contrast with eukaryotic organisms, write in your notes that in a eukaryotic cell, the nuclear envelope, and we mean by the surrounding membrane, nuclear membrane, separates transcription from translation. And then extensive RNA processing occurs in the nucleus. In other words, the nucleus provides a separate compartment for transcription that is missing from prokaryotes. Now also write in your notes that the original RNA transcript called pre-mRNA, here you see it right here, is processed in various ways before it leaves the nucleus as a fully formed mRNA. So you have a pre-mRNA molecule that gets processed into mRNA, totally different than in a eukaryotic cell. And then translation occurs outside of the nucleus. Okay, take a deep breath. That's an introduction to protein synthesis. Way more to come. This is a relatively complicated process, but one that's extremely important, mainly because proteins are so important. So take a picture of those notes, submit them to Moodle, grab that study guide, answer the questions, and submit it. And we'll see you in part 3 of this podcast and part 2 of protein synthesis. Bye.